get license from anti-angiogenic drugs in India in 30-fold less price. I think this is a great news for common people in India, and I hope this will open up a new open source drug discovery initiative made by Government of India. But the question is, what is angiogenesis and what is anti-angiogenic drug? Why it is so important? And what are the current challenges, critical challenges? So I'd like to go over step by step, and maybe I would like to give you some new ideas, new themes. And obviously, it will be a challenge for, pharmacy, particularly pharmacy, who will generate new genetic drugs in near future to really evaluate the BA and BE later part, because it's not that straightforward like common drugs. So, although I'll focus today on the cancer component, because that is my specialty, but angiogenesis targeting is also important for several other diseases, and particularly macular degeneration. It's already one of the top drugs ever has been used to treat the blindness, the old blindness, and it's working, extraordinarily. To go back to the fundamentals, the hallmark of cancer, the cancer already defined by Doug Hanahan and Bob Weinberg 10 years ago. Now they regenerated or rejuvenated the concept. There are several steps to be, a cells or a tissue can be cancerous, and most of the people might know that. But one of the main topics here is angiogenesis. So by definition, angiogenesis is the sprouting of new blood vessels from existing vascular bed, and to be cancerous or to make more metastatic diseases, you need the angiogenesis component has to be happen. And now, based on that hypothesis, Dr. Folkman, like 40 years ago, when he explained that angiogenesis can be an important target to block cancer progressions and metastasis, because cancer cells need those new vascular bed or new angiogenesis due to their supply of food as well as oxygen, glucose and other components, and oxygen, as well as to get rid of the waste products.
More informative for us. And BEGF is one of the major receptors for cancer targeting. You told like that. And neuropilin, one receptor. Is there any specific drugs available to target the neuropilin, sir, like bevacizumab or trastuzumab for EGF?
Well, I just mentioned that Genentech has a drug already, anti-neuropilin antibody. It is in phase 1 clinical trial, different places. But I didn't tell you that we did all the several other work, huge body of work, the role of neuropilin in the, several other organs, like, blood-brain barrier. Actually, neuropilin has a very important role. And I should not comment too much because it is not published and due to some complexity. But I should tell you one thing is, the biology, sometimes we simplified the biology. And as a scientist, as a pharmacist, our job would be to get deep into the truth. And that will help to unveil the lot of problems we face in clinics. So targeting neuropilin is a great idea, but I don't know. I think there will be a lot of other problems will arise, and we have to be careful eventually. Thank you.
Thank you.
Sir, a combination of anti-angiogenic agent and chemotherapeutic agent won't reduce the distribution of chemotherapeutic agent, because anti-angiogenic agents will ultimately reduce the blood flow to the tumor. So, would it reduce the distribution of chemotherapeutic agent?
Actually, just opposite. So, what people found is just opposite. That anti-angiogenic agent helps to make immature blood vessels You know, tumor inside tumor is a highly, the interstitial pressure is very high due to edema, BPF effect. Now, when you put anti-angiogenic agent, now tumor vessels are more mature. That's why drug can penetrate deeper. And since less edema, the availability of drug inside the tumor is much more. So that's why it's just opposite concept. But if you can sustain angiogenic, anti-angiogenic therapy, sustain long term, might not be a great idea. Because brain might be a problem of in survival of normal endothelial cells, and maybe drug cannot be available inside the tumor. Absolutely, you're right. So when you are designing clinical trials, what we are going on right now in our clinics, so when you're designing clinical trials, we have to make sure that we know the anatomy of the tumor of the particular patients. If you don't know the anatomy of the tumor of particular patients, we don't know how long we need to treat, what will the follow through, and also when treatment is going on, what kind of experiments or tests we should do to make sure to raise the question, how long really anti-angiogenic therapy should go. So that's why we get to make sure about those kind of experiments. Thank you.
Do you think that there's a problem using a model where you look at an effect in mice and maybe you prolong the lifespan of the mouse by 1 or 2 months, and then you go into the clinic and you're expecting that it's going to prolong the lifespan in the human by 10 to 15 years? This is what I call the 30-day tumour effect. You study it for 30 days, you write the paper up, and that's your advancement. Instead of what we used to do, which was the 250 to 365-day tumour effect. If you look what happens long time, and then see.
Well, mouse lifespan is only 2 years, and human lifespan 80 years. So if you compare that, there is like 40-fold difference, So we can expand that 40 times.
I think you could say that if you're talking about the relative lifespan of a dog and a human, but when it comes to the tumours, I think you're pushing it uphill a little bit.
I know. Put other way around, so far, there is no other good model system or tools we have in the laboratory to do the preclinical trial. That's why still, the mouse model of tumor is so popular. Now, I agreed with you. I showed you that when you treat pancreatic cancer in the rapamycin Hyderabad combination, now it is clinical trial. It's fabulous in pancreatic cancer system in mouse, but miserably failed in the human patients. So this is not only lifespan. I think again, anatomy of the tumor development, in microenvironment, there are several factors there, and we cannot mimic in the mouse model as such. But to say so, I believe there is some effectiveness still there in mouse model. So I cannot exclude everything. The mouse model is bad. There are something good, but we need to be better.
Maybe we should be starting on tumours that are already 1.5 grams in a mouse, rather than standard tumour models to start with something that's 100 milligrams. And a 100 milligram tumour in a human would not do anything.
Oh, I agree with you, because there are, I think most of the work that's right now going on, put other way around. I mean, most of the animal experiments done so far in laboratory, putting implanting tumor as well as drug same time. And that is, you can see so many papers publishing like that. But real challenge would be that get the tumor as mimicking as human disease. In more extravasant, mostly like a pancreatic cancer, make the pancreatic cancer as such before you start that already metastatic diseases and is an unresectable tumor that we see in clinics. And then you start the experiment. And that's exactly the trend going on in that direction. So most of the paper I can see, good journals are working like that. But even say so, I don't know. I mean, can we really mimic human disease in the mouse? I don't know, but at least we get something there. You shouldn't develop it in public. Same model, exactly same model, much faster. See, one of the challenge, I'm sure you do know, that if you make a spontaneous tumor model, in mouse with immune compromised mice, immune competent mice rather, so it sometimes takes longer time like 4, 5, 6 months to grow the tumor. And that's not always good for therapeutic model. So what we did so far, I mean, very recently we didn't publish it yet, a completely new model, a spontaneous pancreatic cancer tumor model within 6 weeks. Animal died.